pleased to share with you some technology we're working on to solve the problem of vaccines against RSV. We've worked on this for many years and we failed over and over again, but we have a new approach that we think may actually work. So briefly, I'm going to touch base on RSV and why classic active vaccination hasn't worked. I'm going to touch base about our molecule and why we think it may solve the problem. A bit about the clinical testing we've done with our molecule, and then how we plan to use it in the north and the far south, and then as well as the alternate strategy to use for global access in the tropics and in impoverished countries. So RSV is ubiquitous. Everybody gets it every year. Half of all infants will get it by their first year of life, and 100% are infected by 2 years of age. And infections are common throughout life. We all get RSV every year, likely. For us, it's a cold. But when infants get it for the very first time, they get lower tract illness and hospitalization. And during winter season, 90% of all bronchiolitis, lower tract illness, is caused by RSV, and 40% of all pediatric pneumonias are caused by RSV. There's also an emerging link between RSV infection when you're an infant and asthma as you get older on, and that's still being teased out in the literature now. We do know that neutralizing antibody to the F protein, which is the major surface glycoprotein of RSV, is protective and can prevent disease and hospitalization. And that's the basis of palivizumab, which brand name is Synergis, which is marketed around the world to prevent preterm infants from getting ill from RSV. Here's a recent global view about causes of death around the world. On the left-hand panel, you have between 0 and 27 days, and the right-hand panel, you have between 1 month and 1 year of life. And you can see that the major cause of lower tract illness, death due to respiratory illness, is in fact RSV. So it's a global important illness, and I think it's recognized by everyone who's ever been in the clinic during the wintertime. Now here's the problem with developing a vaccine for infants. The vast, which you see on the bottom, is the months of age, and the other axis is the percent of hospitalizations caused by RSV. And you can see the majority of the hospitalizations occur in the first, really 6 months of life, with a spike being the first month to 2 months of age. And in fact, by 6 months of age, roughly half of all hospitalizations has occurred. So to develop a vaccine where you can have active immunity in those first 6 months is very difficult indeed, especially if you think a multiple dose regime is needed. So maternal vaccination is being pushed right now as a way to do it, vaccinate the mother and have passive transfer of antibodies to the infant to protect during that age. An alternative is to develop a passive vaccination, which is essentially giving the antibody directly to the newborn. And that's the approach we're taking on. So we call this MEDI8897. This is a super high potency monoclonal that's derived from human B cells. It has our half-life extension technology baked into it, and we can make this at very high efficiency in fermentation. And this allows us to give a once per season dose with a fixed IM dose. It's not a weight-based product. And we're striving for vaccine-like pricing so we can make it available to every child in the world. We're looking to prevent a lower tract illness and severe RSV illness. And the indicated population will be term infants as well as preterm infants. For those at extra high risk, those children with chronic lung disease and cyanotic heart disease, they may perhaps get this drug the first and second year that they are alive. And we've completed the adult studies, and we're now in preterm infants, and I'll touch on these studies a bit later on. So here is the target. This is the F protein of RSV. Our drug binds the pre-fusion conformation of RSV-F, which is distinct from palivizumab. And it's a fully humanized IgG. It doesn't have any mutant sequence at all. And it targets the site 0 sequence, which is unique and is now, well, relatively recently it's been crystallized, so it's well known now to those people who work with crystals. Here's some data that compares our molecule versus palivizumab. What you have on the right-hand side is RSV-A, on the left-hand side RSV-B. In purple you have Pali, and in green you have MEDI8897. You can see that it's roughly 2 orders of magnitude more potent than the drug which is currently licensed. And it neutralizes all RSV-A and RSV-B viruses we tested. And you can see the same effect when you look at in vivo. So these are cotton rat models. On the right-hand panel, you have RSV-B, on the left-hand panel, RSV-A. And what you have along the bottom is the amount of antibody, and along the top is the amount of virus recovered after challenge. And you can see a nice shift to the left with MEDI-897, which is the one in green. It's roughly an order of magnitude more potent in vivo. And it allows us to calculate EC90, which is the neutralization of 99.9% of viruses recovered from the cotton rat lungs. And that becomes actually our target concentration, which we're shooting for in the clinic. So a bit about our YTE technology, and for that, let me get into a small amount of science. The major degradation pathway for antibody is there's taken up in the endosome. It binds to, or doesn't bind to the Fc-RCN, which is a fetal Fc receptor. And if it doesn't bind, it gets degraded like across the bottom side. So what the YTE is 3 mutations in the Fc region of our monoclonal, and it increases binding to FcRn at acidic pHs. So it binds tightly within the endosome and gets recycled back out into circulation so it doesn't get degraded. And this, in our previous experience, has increased the half-life of monoclonals that we put into people by 3-fold. I'll show you some human data on that in a second. So here's a model we generated. The green line is the EC90, which represents 99.9% reduction of virus in animals. And what you have are 3 independent models. In orange, you have one for a 6-month-old baby by weight, in blue by a full term, and in purple by a pre-meat with 95% confidence intervals. And you can see according to this model, even for the heaviest babies, those that are 6 months of age, they maintain levels of the drug above the EC90 up to 150 days, which is 5 or 6 months, which is the length of the northern and southern RSV season. So we took this into the clinic in adults. This was a study of 136 adults, which was placebo-controlled. And we tested 3 different dose levels up to multi-gram doses, and I'll show you that in a second. And we followed them for a year. And basically, the drug was extremely safe, as we expected. There was only a single SAE, which was that of trauma. The bioavailability was 85% for the intramuscular doses, and the half-life was extended between 69 and 77 days. The normal half-life for immunoglobulin is 21 days, so we saw the expected prediction. There was no anti-drug antibody, and there was no impact on PK for those that did have a small amount of ADA. So here's the human data. It looks like we drew these lines, but this is real human clinical data. The bottom 2 are intramuscular doses, and the top 3 are IV doses. And this is the data that allowed us to calculate the serum half-life of 69 to 77 days. So this confirms what our model predicted, hence we still believe that a single dose will protect for the entire season. So we're currently in infants. This is a study which is currently vaccinating in South Africa and Chile. And we're testing 3 doses of intramuscular antibody, and we're looking at the parameters listed. So far, there have been no safety issues, and we still have yet to unblind the PK on this drug in infants. So how would we plan to use this drug? In the north, in Europe, North America, and Japan, there's seasonal administration is planned, same in Australia and possibly Argentina and South Africa. And we would give this vaccine when the infants enter their first winter season. So come November time, they would get a jab. It would protect them for when they risk exposure. It would be a single fixed IM dose. But there's obviously a problem. Here's a recent review of Epi globally. And you can see that in the north and south, a 5 or 6 month period would nicely cover the epidemics that are seen globally. But in the tropics, both in the northern and southern tropics, the epidemiology is irregular, is uncertain, and sometimes moves around quite a bit. So for this, we're proposing an alternate dosing strategy where we would dose infants as they're born as birth doses. And this would provide protection for the first half year of life, where the risk of serious disease is greatest. If you remember the histogram, most of the disease did in fact happen in the first half year of life. And this eliminates the need for RSV surveillance, which is sometimes tricky in less wealthy countries. And it takes advantage of pre-existing medical contacts so they can be vaccinated at birth without having to schedule a seasonal vaccination. We are proposing to, or we're planning on pursuing this in parallel with the development we're doing in the north, so that this drug can be available globally at the same time, both in the north, in the south, in the tropics, as well as temperate zones. So why do we think giving monoclonal at birth to a term infant would actually protect these infants at all? This isn't the way this module is used before. We had previously conducted a study in Native American term infants where they received 5 monthly doses during the winter time. This was a pretty large study of over 2,000 infants, and they were young when they were aged. But it was given during the winter, like palivizumab is given currently. And what we saw was a 83% reduction in hospitalizations and a 71% reduction in outpatient lower tract illness. So this, in our minds, gives us some evidence that a passive vaccine given to term infants will protect against lower tract illness as well as hospitalization. Here's a couple of graphs that show that. That are from the publication which will be coming out soon in Lancet Infectious Disease. Once again, hospitalization on the left and outpatient lower tract illness on the right. We also know from experience in Taiwan that giving it in birth doses where transmission is variable works as well. Taiwan has a blend of year-round transmission and twice yearly RSV epidemics. In this study, they gave 6 monthly doses when the babies were discharged from the hospital. These were all preterm infants. And they compared the rates to historical RSV hospitalization rates. And what they saw was that for the first 6 months, they saw an 86% reduction in hospitalizations. This was irrespective of seasonality of RSV. And for the first 12 months, they saw a 78% reduction. So taken together, this gives us multiple lines of evidence that using a passive vaccine in these term infants will save hospitalizations as well as prevent serious disease. So this just recapitulates how we plan on using it as a passive vaccine, as a birth dose to protect for the first half year of life. And once again, a single IM dose is planned. So overall, we think we have a validated target and a validated approach with a passive vaccine to meet this unmet medical need. This is modern monoclonal technology. This isn't the monoclones we used in the past. And it allows us to go for once per season dosing and use a tiered vaccine-like pricing to be able to make this accessible to the entire world. And we see great opportunity in doing parallel development, both in developing as well as developed countries. This is an approach which has been supported by IVR WHO, and we're in discussions now how to pre-qualify a passive vaccine, a monoclonal, and that seems to be moving pretty well. From the FDA perspective, we have fast track designation, and they've looked at our clinical endpoints as the EMA, and this seems to be well on its way to being approved at this stage. So thank you for your time.
So questions? If I could lead off, Philip, I mean, this is obviously very innovative in its approach. Do you see this being extended to other disease targets, and are you currently looking at other disease targets where you may be able to passively immunise?
Right. This would work best for infants because of their size. When you get to be adult size, you need too much antibody, it becomes less useful. But you could think along the terms of pertussis, perhaps, or influenza. Those are both diseases that attack in the first half year of life where this could be useful. We have programs in influenza, but it's more on treatment and less for prophylaxis, where they can be repurposed.